Have you ever wondered how your body knows exactly how to build, well, you? To find out, we have to go deep inside your cells, right into the center, a secure vault called the nucleus. Inside this vault sits your DNA. Think of your DNA as the ultimate master hard drive. It holds the original source code for every single thing your body needs to survive and grow. But here's the problem. The factories that actually read this code to build things, like muscles, enzymes, and skin, are located completely outside the nucleus, and your DNA is way too valuable to leave the safety of the vault. To solve this, the cell does something simple. It makes a temporary, disposable copy of just the specific instructions it needs at that exact moment. We call this biological flash drive RNA, and the process of downloading that code from the DNA to the RNA is called transcription. By keeping the DNA safely locked away and only sending out these temporary RNA copies, the cell ensures the master source code stays protected from any wear and tear on the factory floor. To make that temporary copy of the genetic code, the cell uses a highly specialized molecular machine called RNA Polymerase. This diagram shows the transcription unit, the specific DNA segment targeted. The promoter on the left is a start button, telling the machine exactly where to dock. Notice the two DNA strands. The machine only reads one specific side, the template strand. Having these specific start buttons ensures the cell only spends energy copying the exact instructions it needs right now. Without them, the cell would waste its entire energy supply copying millions of useless genes it doesn't even need. Once docked, the machine moves forward. It physically unzips the double-stranded DNA helix, exposing the hidden code inside to be read. Why copy only one side? If the machine copied both, the two new RNA copies, being chemical opposites, would immediately snap together. They form a solid block the factory can't open, permanently trapping the instructions and making the file useless. This diagram shows the complete cycle. Once the copying machine reaches a specific stop sign sequence on the DNA, called the terminator, it simply pops off, releasing the finished RNA copy. The cell now has the data it needs. But like raw camera footage, this newly downloaded data is a bit messy and needs to be edited before it can be used. For complex creatures like us, the newly created RNA flash drive is actually full of junk data, sections of code that aren't part of the final instructions and would confuse the factory. Here, the code splits into exons that exit the nucleus and introns that stay. The cell uses molecular scissors to snip out introns and glue exons together, a process called splicing. Finally, it adds a protective cap and tail to secure the file. Splicing and capping is like editing a film and placing it in a secure case. The data is now polished and ready to be shipped. Our finished RNA flash drive finally slips out through a tiny opening in the nucleus wall and enters the main body of the cell. We just watched the cell find the start button, unzip the master code, copy a single side, and carefully edit the result. Now that the instructions have arrived safely, the cellular factory can begin 3D printing a protein based on that code, a process called translation. This microscopic data transfer is happening millions of times a second inside you right now, keeping you alive. Stick around, and next time we'll see how your cells actually read this code to build you from scratch.